I think we should start with the most important question. What is a marine protected area? Commonly called MPA, it is a specific area reserved by the law to protect the designated environment. They have been established to protect vulnerable species and ecosystems, to avoid negative anthropogenic impacts, and to enhance the productivity of fish and marine invertebrate populations in the restricted area. According to the National Marine Protected Area Center, MPAs could serve as a source of baseline scientific data to monitor the changing solutions in the marine environment. They can also serve as educational destinations and provide opportunities for educational dives. Other benefits can include the overall idea that they do provide a safe habitat for corals and endangered marine life. One study done shows that population biomass, the size of fish, and the density in a given area and species diversity has increased in marine protected areas. However, there are many conflicts within communities because fishermen rely heavily on the fishing industry to get by, so they don't agree with the idea that you cannot fish within MPA boundaries. However, what many people don't understand is that MPAs allow the fish to be replenished, their population will grow, and eventually the fish will wander out of the areas and into the areas that can be fished. A study done by a group of researchers, including Florida Tech's very own Dr. Ralph Turingen, compared the growth rates and asymptotic length of fishes between MPAs and fish reefs in the Philippines to provide evidence that MPAs promote larger fish and more sexually viable fish populations than non-protected areas. The researchers revealed that the asymptotic length, or the average length of fish of a given stock can reach if they were to grow for an infinitely long period, as well as the growth rates of fish differed between MPAs and fish reefs. The populations within MPAs observed larger lengths at 50% maturity compared to nearby non-protected areas, meaning fish of the same age were larger within MPAs than outside of MPAs. The results imply that MPAs protect against impacts of size-selective harvest, which is the targeting of the largest fish by fishermen. A favorable phenotype is a physical expression of a gene that provides an organism with the most appropriate tools to survive and reproduce in a certain environment, such as camouflage. A favorable phenotype for fish is reaching sexual maturity at a larger size because the larger fish are able to produce larger, more viable larvae than smaller, sexually mature fish. Shifts toward this favorable phenotype were most common in the oldest and largest MPAs, but occurred in all of the MPAs examined by the researchers. However, size-selective harvest causes fish in non-protected areas to assume unfavorable phenotypes, meaning the largest fish are removed by fishermen, causing the smaller fish to become sexually mature at smaller sizes in order to continue the population. So, to recap, this study found evidence that MPAs in the Philippines promote larger, more sexually viable fish, and non-protected areas do not. This study and many others show strong supporting evidence that marine protected areas do in fact have a positive effect on marine ecosystems.
Our project idea. The scientific literature, as mentioned before, shows supporting evidence that MPAs are safe havens for both corals and fish. Thus, our project focuses on the abundance of live and dead corals and the diversity of fish in and out of the Tres Palmas Marine Reserve in Rincon, Puerto Rico. We sampled 3 locations of fish diversity, 2 of which were within the boundaries of the MPA and 1 outside. The 2 MPA sites are known as Tips and Keyhole. The non-protected area is known as Marias. Likewise, we sampled corals within and out of the Tres Palmas Marine Reserve. The samples collected can provide supporting evidence that MPAs protect corals from natural and human-induced impacts and promote fish diversity. Hypothesis and reasoning. Since there is strong evidence that marine protected areas inhibit harmful activities such as fishing, anchoring, and collecting marine life, it is inferred that the MPAs will observe a higher percentage of live coral and a greater diversity of fish than non-protected areas. Methods of collection. a 25 plus meter tape measure, a PVC quadrant, a slate to write down data, an underwater Olympus camera, Microsoft Excel, and Coral post-processing software known as CPCE were used to collect data, analyze the abundance of coral, and quantify fish diversity in and out of MPAs. The tape measure was pulled out 25 meters in a straight line parallel to the shoreline and laid across the benzos, or ocean floor. The necessary information was recorded on the slate, such as time, location, water clarity, depth, etc. Every 5 meters, the quadrant was placed above the tape line, and a picture was taken of the quadrant with the ocean floor beneath it. The CPCE software was used to randomly select 20 points on the images of the quadrants. At each of these points, it was the researcher's job to identify what object the random point landed on, such as live or dead coral, macroalgae, or even the tape measure itself. The CPCE software essentially allows us to sample the percent coverage of live and dead corals on the reef through the use of pictures. Furthermore, another 25 plus meter tape measure was pulled out to 25 meters, and the fish 2 meters to the side and in front of the researcher were recorded on the slate as the researcher swam the 25-meter length. Information such as the species of fish, number of fish, and size of fish were recorded on the slate. However, for the purposes of this experiment, only the number of species and number of fish observed was used. If an unknown fish was observed, the researcher would record identifying marks, such as a certain pattern of colors or a unique shape, and identified the fish later. The Shannon-Weiner. The Shannon-Weiner index measures the diversity of a population by taking into account the number of species in a population, or species richness, and the relative abundance of individuals within each species, or species evenness. Through a series of simple calculations involving the ratio of the sample population to the total number of fish observed, the logarithm of that ratio, you can obtain a value of Shannon Diversity Index. The Shannon Diversity Index measures the actual species richness and species evenness, which together show species diversity. A higher value of the Shannon Diversity Index equals a more diverse sample population.
For the first part of our experiment, we conducted a survey of the abundance of live and dead coral in and outside of the Tres Palmas Marine Reserve in Rincon, Puerto Rico. The measure of central tendency or average used in the experiment was the median or middle observation recorded because the data did not meet the certain criteria that would allow us to use the mean, which makes use of all of the data. That being said, the graphs on display are called box plots, and they show us the median abundance of coral as the darkest horizontal line, and the largest and smallest observations as smaller, lighter horizontal lines. The box plot on the left represents the abundance of live coral in and out of the MPA, and the box plot on the right represents the abundance of dead coral in and out of the MPA. As you can see, the dark lines representing the median coral abundance is different between the MPA and non-MPA, so the question that needs to be answered is whether the median values are far enough apart to be considered statistically different. To do this, we conducted a variation of a t-test called a Wilcoxon signed rank test because the data did not meet the criteria for a t-test. The Wilcoxon test tests if the median abundances of coral are far enough apart to be considered statistically different. The tests revealed that the abundance of live coral in and out of the MPA was statistically similar. In other words, the amount of live coral seen within the boundaries of the MPA was statistically the same as the abundance of live coral seen outside of the boundaries of the MPA. Furthermore, the Wilcoxon test revealed that the abundance of dead coral inside the MPA was significantly different than the abundance of dead coral outside the MPA, with the non-MPA site observing a higher abundance of dead coral. In short, this means the non-MPA site had a lot more dead coral than the MPA site. So, in conclusion, the MPA site had the same amount of live coral as the non-MPA site, but the non-MPA site had significantly more dead coral. These results partially agree with our hypothesis that the MPA sites would have more live coral and less dead coral. A likely explanation as to why the two locations had the same amount of live coral would be that the two locations are subject to the same environmental conditions, such as surges caused by hurricanes. The non-MPA site may have had more dead coral because, although the two locations are subjected to the same environmental conditions, they are not subjected to the same anthropogenic or man-made impacts. As mentioned before, MPAs prohibit anchoring and any collection of marine life, including fish, or non-protected areas do not. So the higher abundance of dead coral outside of the MPA may have been a result of laxer regulations than the MPA sites. So, as mentioned before, we conducted fish surveys at 3 different locations in the same marine reserve in Rincon, Puerto Rico, and compared the average Shannon-Weiner diversity index values between them. The 2 MPA sites were Keyhole and Steps, and the non-MPA site was Maria's. The measure of central tendency, like the coral data, was also the median. Similarly, the box plot shows us the median Shannon-Weiner index value as the darkest horizontal line, and the largest and smallest observations again as smaller, lighter horizontal lines. The median diversity index values are different at each location, with Steps observing the largest median value, Keyhole observing the smallest, and Maria's somewhere in the middle. So the looming question that needs to be answered is whether the median values are far enough apart to be considered statistically different or close enough to be considered statistically similar. To do this, we ran a variation of an analysis of variance test, or ANOVA, called a Kruskal-Wallis test that tests if the median values are the same or different between the 3 locations. We also conducted a Dunn test to interpret the results of the Kruskal-Wallis test because the Kruskal-Wallis test only tells us if the median values are the same or different at each location and fails to explicitly specify which locations are statistically different from each other. The statistical tests revealed that the 2 MPA sites, Keyhole and Steps, had significantly different median diversity index values. However, the non-MPA site, Maria's, did not have a significantly different diversity index value from either MPA site. That is to say that Maria's did not have the lowest diversity as expected, and the MPA sites were statistically similar to the non-MPA site in fish diversity. We hypothesized that the MPA sites would observe a higher diversity of fish, however, that is not the case as indicated by our data. There are a number of explanations as to why our hypothesis was incorrect, including the small sample size and a short sample duration.